Okay, class, we're using two PowerPoints. One is the Fox PowerPoint that I modified, and the other is the Mari PowerPoint to discuss fertilization, pregnancy, and delivery, which is called parturition. So we'll start over here first, and I'll say that pregnancy are the events that occur from fertilization until the infant is born, parturition. Conceptus is the developing offspring. The gestation period is from the last menstrual period until birth, and we'll discuss this again. Okay, so we have what we call the pre-embryonic period. Many will use the term the pre-embryonic period, which is the first 16 days after fertilization. And during that time, the reason they call that is you're undergoing something we'll call cleavage, which really is mitosis, implantation into the uterine wall, the stratum functionalis, and embryogenesis, the beginning of the development of the actual embryo. The embryo conceptus, the embryonic period then, the embryonic period, if this is the pre, then would be the conceptus during the 3rd through the 8th week of conception. And from the 9th week of conception through birth is called the fetus. Okay? So looking at it again, conception is actually fertilization, sperm meeting egg. That's the time from fertilization. The first cell you get is called the zygote, when sperm meets egg in fertilization, till delivery. That time is about 266 days, or if I do the division, 7 into this, 38 weeks. But everything, remember, in clinical is a range, so 36 to 40 weeks. Gestation, the term gestation versus conception begins with the LMP, the last menstrual period. Okay, and so therefore, since it starts at the last menstrual period, and the beginning of the last menstrual period is the first day of the new cycle, then you add 2 weeks to that, to the conception, because the female is not even pregnant, because the female is not even ovulated. So that puts the gestation period as 36 to 42. Now, when a female that thinks she's pregnant goes to the physician, they try to get an idea of when is the pregnancy going to be. So they will use what we call the LMP, the last menstrual period, because obviously the female may not know exactly when she got pregnant. So the last menstrual period then will be used, and a wheel generally is used. It kind of looks like this to calculate the EDC. The EDC is called the estimated date of confinement, the day she's going to deliver, basically. That's where the confinement. And that's approximately 40 weeks, with a range again of 38 to 42. If the lady delivers before 38, we say she is predates. If she delivers past 42, she's post-dated delivery. Now, the problem with the past 42 is the placenta itself only has a certain time span. So fetal distress will start to occur after 42 because the placenta is trying to give out. Okay, stop functioning. All right. So right here would be kind of looking at the relative size. Fertilization, zygote, first, and it just kind of keeps going until you form the embryo, which would be about the 3rd week, something that looks humanoid, I guess you could say, and so forth. All right. And so we'll go here now. Let me go to this one. So about 300 million sperm enter the female at ejaculation. Now the sperm are being stored in the male epididymis, the seminiferous tubules where they were formed. Only about 100 of these live to enter the fallopian tube, the oviduct. In order to fertilize the ovum, a sperm must become capacitated. Really, that means activated. That takes about 7 hours in the female reproductive tract after ejaculation. So capacitation occurs in the female reproductive tract. Things that happen is in the epididymis, to keep the sperm kind of inactive, in a resting stage, the pH is about a 6.5. That's somewhat acidic. So then hyperactivation of the flagellum is due to progesterone opening of cat spur channels, and the capacitated sperm are guided to the ovocyte by chemical attraction and thermotaxis. So in other words, again, you're keeping the sperm inactive, then ejaculation occurs. In the female reproductive tract, the pH is somewhat alkaline to bring the pH up to become more alkaline and activate. And then also some things are going to happen, particularly with calcium and so forth, to activate the swimming flagella, this flagella, and then how it's guided into the fallopian tube towards the ovum. The ovum is going to be chemical attraction and some heat attraction. Okay, let's go a little further. So I'll go here. The oocyte is viable for about 12 to 24 hours after ovulation. That's what this means. The sperm is viable about 24 to 72 hours after ejaculation and in the female reproductive tract. For fertilization to occur, intercourse must occur no more than 3 days before ovulation and 24 hours after ovulation to account for both of these time spans. And remember, that was the rhythm method in contraception, because intercourse before 3 days or after the 24-hour period would rarely cause a pregnancy. Okay, fertilization is when the sperm fuses with the egg to form the zygote. Okay, we'll go a little further. The fates of the ejaculated sperm, remember going back that 300 million sperm entered there. So in other words, the fates of the ejaculated sperm, they leak out of the vagina immediately after deposition. So some just leaks out. Some is destroyed by acidic vaginal environment. Okay. The vagina itself is somewhat acidic to prevent overgrowth of certain bacteria, because there's certain bacteria. But once ejaculation occurs, those that can get into the cervix, into the cervical area, that's where you'll encounter the alkaline material. But in the vagina, it's more acidic. Fail to make it through the cervix, which I mentioned, dispersed in the uterine cavity. So some that goes up there will not make it through the cervix. They just go around the cervix into what we call the fornices, something I brought up in the anatomy area. The dispersed in the uterine cavity are destroyed by phagocytic leukocytes and then reach the uterine tubes. Sperm must undergo capacitation before it can penetrate the oocyte. So in other words, now we're back to capacitation again. Okay? So I go further. So the capacitation involves acrosomal reaction and sperm and to get the sperm activated. Okay, so let's use this PowerPoint for that. Capacitation involves 3 important chemical changes. A change in pH. Now, capacitation is, again, to activate the sperm that's in the female reproductive tract. So we have to change the pH. Calcium concentration becomes important, and cyclic AMP. Cyclic AMP. Okay? It takes about 7 hours in the female reproductive tract where the pH is alkaline, but again, that's in the endocervical area. The female vagina itself is more acidic. In the female reproductive tract, the sperm exchanges H+, which is acid, for sodium, cation for cation, as it enters. The sperm cytoplasm pH changed because, so in other words, the H+ is ejected out of the cytoplasm, okay, of the sperm, which would make it less acidic in there, and sodium replaces it to keep the electrochemical balance. The sperm cytoplasm pH change to more alkali activates the dynein proteins. Now go back and study flagella. Dynein proteins are the proteins that actually move the flagella. The flagella has microtubules, but they must be moved by dynein proteins, activating them and allowing the sperm to move. Okay, so we go a little further. As the sperm gets closer to the oocyte and the female oviduct, fallopian tube, progesterone released from the ovary along with the oocyte activate calcium channels in the sperm flagella's principal piece, causing the hyperactivity. The principal piece in the sperm is that area we discussed before, where that's the beginning of somewhat of the flagella tail. Okay. Highly activated sperm are guided towards the ovulator by chemotaxis and thermotaxis, a warmer temperature. Okay, go a little further. Okay, so now we'll come over here. So an ovulate is encapsulated. Remember, we talked about this. What is ovulated is the oocyte in metaphase 2. It is surrounded by a glycoprotein zona pellucida and some granulosa cells known as the corona radiata. Okay, the sperm binds to the zona pellucida. So the sperm is going to try to bind, let me see if I can find a picture, to the zona pellucida here, and undergoes the acrosomal reaction. The acrosomal reaction in the sperm is this cap here, which is a vesicle containing enzymes that can dissolve through these cells here, corona radiata, and the zona pellucida. So sperm binds and undergoes acrosomal reaction. Enzymes are released near the oocyte. Hundreds of acrosomes released. These are vesicles. Okay, so I come back here then. Here we go. Fertilization, let me be sure we are okay. Fertilization usually occurs in the fallopian tubes, generally in the ampullary area. The acrosome is a vesicle inside the cytoplasm sperm at its head. There's a surrounding cell membrane separating it from the acrosomal membrane. Okay, so you have a membrane, and then within that, just like any vesicle, two membranes, the cell membrane and the membrane of the vesicle. When the sperm binds to the zona pellucida, calcium channels are opened in the sperm head. Okay, this calcium entry allows the membrane, and this is basic exocytosis from earlier activity that we saw early in the course in ANP1, to fuse with the cell membrane of the sperm, leading to exocytosis of the acrosomal contents. Proteases, which would be enzymes that develop proteins, and hyaluronidase, which is a glycosaminoglycan, dissolves the zona pellucida. That's what it's made up of. And these enzymes are released from the acrosome. Okay. So again, the sperm binds to the zona and undergoes acrosomal reaction, which we just talked about, and enzymes are released near the oocyte. Hundreds of enzymes are released, and that's where we are now. Okay, let me go here. So I go here now. Okay. Once a sperm makes contact with the oocyte's membrane, okay, that means it's gone through the corona radialis, and he's actually making contact with that. Beta protein finds and binds to receptors on the oocyte membrane. Alpha protein causes it to insert into the membrane. So I'll come here. So we're trying now to see how we're going to get it to lock into that. Okay. And now we're going to come back again to the acrosome reaction. So I'm going to close here and then start right here at